Enzootic nasal adenocarcinoma. You can say new disease because no comprehensive reports on this disease are in India. And previous year, we encountered two outbreaks of ENA in Tong district and Jaipur district. This ENA is caused by enzootic nasal tumor virus, ENTV, and it is genetically related to JSRV virus. And there are two types of virus, ENTV1, which cause neoplasm in sheep, and ENTV2, which cause neoplasm in goats. This disease was first reported in 1980s. After that, it is observed in some countries, except Australia, New Zealand, and United Kingdom. In India, as I told, there is no comprehensive report and there is no confirmation of ENTV virus from sheep or goat. Generally, prevalence is ranging from 0.1% to 0.3%, and sometimes it is very high, and it will go up to 15%. And no genetic, sex, or breed predisposition has been reported in this disease. I was talking about the outbreak of this ENA in Tong district and Jaipur district. So total 600 sheep were affected, and there was 10 to 12% mortality, and morbidity was around 20%. First clinical symptom is that signs is that unilateral or bilateral protrusion of eyeball. Then these eye lesions were very extensive with loss of natural reflexes and loss of vision. Glaucoma is there. Then mouth breathing, frothy salivation. Sometimes the animal is circling, and there is a rapid muscle wasting is there. And we found that the location of the tumors was in olfactory mucosa of ethmoid cartilage, and this growing tumor compressed upon the walls of nasal cavity and and this protrusion of eyeball is resulted. Here you can see the protrusion of eyeball, unilateral protrusion of eyeball, and there is complete loss of vision, no reflexes at all. In another case, you can see right eye is protruded in comparison to left eye. From some sheep, we also collected the nasal discharge, and that discharge we stained by May Greenwald Jimsa stain, and we find that these tumor cells means clumps of tumor cells, and on the basis of that we diagnosed it as nasal carcinoma. We conducted some 2-3 postmortems. Those sheep we got died in the field, and we conducted the postmortem. After opening these turbinate bones, this nasal cavity is found occluded with tumor. You can see the tumorous growth here, and in this ENA, it is reported that there is no metastasis in any organ. Only the tumors are limited to the turbinate bones. But we found that in lungs also there is tumorous mass. You can see here. This is cut surface with necrotic debris and bloody exudate. We process these samples for histopathology. In lungs, we find the similar lesions like OPA. These alveoli with papillomatous proliferation and infiltration of mononuclear cells giving adenomatous appearance, similar like OPA. Then in lung, these nasal tumors, we find similar changes like lungs. These cases were confirmed in the laboratory by GAG-gene PCR. This PCR we standardized in the laboratory, Also, we standardize real-time PCR and identify this infection. So thank you very much.